Thanks for the invitation. So let me start by saying a few words about my background. So my work is mostly in probability and computer science, and often on connections between phase transitions in physics and algorithms, and sometimes touching on information theory, and also on probabilistic graphical models and causality. The closest I've ever come to all of this was a euphemism to say that I'm not a biologist who is in the unenviable position or enviable position, I don't know, of giving a talk about biological problems. The closest I've come to biology before this is doing some mathematical models of evolution. So, but let me start nonetheless. So the problem I want to look at is about the structure and dynamics of certain biomolecules, in particular RNA. And this is a project that we have recently started with Sachi, who's at NCBS, and Hari, who is also in TIFR Mumbai. So the problem here is sort of well known. It has been mentioned already a few times in the talks before this. And it's about going from a sequence to a 3D structure. That's usually the more famous version of this is for proteins. So you have amino acid sequences and you want to go to the 3D structure of the protein. The version we want to look at is where you have nucleotide sequences. So just 4 letters in the alphabet rather than the 20 odd for proteins. And you want to go to a 3D structure. The questions that you want are similar as the protein case. So you want to find what the final structure is, the stable, somewhat stable structure that comes from a sequence. And the second one is this, which I'll come to later, which is you want to also know how this stable structure is actually achieved. So somehow there is a molecule, there's a big molecule. It starts off in some configuration, but it slowly comes into this final structure. The question is, how does it come to that? It doesn't sort of start off there. This will come to later. So here is just an example of what such a 3D structure might look like. So this is for a very small RNA fragment. It's actually from 2001, the structure. So you can see it looks a little bit complicated, but maybe now it'll look a bit simpler. So this is actually the RNA chain. This is what corresponds to the sequence, and the other things sort of attach onto it. So we can talk about more detail about this. So why should one do this? So we've all heard about AlphaFold already multiple times, and the thing here is a more recent result which I found quite interesting. So here, so let's forget about the machine learning thing. So what they did really is this. So they had a neural network. The neural network existed 2 years before that. They had a neural network which takes not the sequence, but actually the structure of a protein, of a small part of the protein as input. And it tries to do somewhat like an inverse problem. It tries to see which are the viable amino acids that can inhabit that local environment. And that lets them know what are the viable mutations they can make to a particular structure. So that's just one part of the thing. Then using this, so they could whittle down to which are the viable mutations and get a mutation which does much better at eating plastic. So it's an enzyme that eats plastic, but it was achieved through finding these viable mutations, which is from a sort of a neural network that is solving the inverse problem of this, going from structure to one location and saying, what can you change this? Okay, so and this neural network it's not, as things go, it's not that complicated. It's big. There's 4 so-called 3D convolutional layers, 3 fully connected, but it's nowhere like GPT or. So for RNA, it's different from proteins, and there are many reasons that make this problem different. So one is that there is much less data. So this is just a numeric comparison. This is actually the last time I checked 2 days ago. About 190,000. That's like 2 lakh protein structures, and just 6,000 odd for RNA. And the other thing is that for the thing you want to do, making this backbone structure, RNA structure is far less constrained. So proteins, because of the nature of the covalent bond involved, there are many more constraints on what the backbone can do. Famously, the Ramachandran plot captures some of those. There's far fewer constraints in the RNA world, and so the number of sort of local angles you need to keep track of is much larger. So here is what has been done for RNA. So here's another recent paper. So it again it takes a sequence, puts it through some neural network, and gets these torsion angles per site. But let's see what it does. So they don't say how many parameters they have. I tried to put it into TensorFlow given what they were describing. It's about 30 million. Again, not that big by these standards. But the funny thing is that It's trained on about 300 sequence of median length 80. So it's not a lot of data. So the training time was probably not that high because there's not too much, that's not enough. There's so much data to run on. They get decent results based on just this. The network was not too complicated. I didn't mention what it is here, but it's yeah, it's again, it's inspired from these language models, but it's less complicated than the mute compute. Okay, so before I go further about what we want to do, there's I think this has come up in this thing. So one important concern I have, at least whenever I see machine learning applied to science, is that is it a case of this, that there's a hammer and we want to put, and we have a nail and we want to put it. But I would like to say that maybe it's slightly different. It's like it's somewhat more principled. There are many hammers and there is a particular nail, and you want to see which one is the best one, and you want to do that both theoretically and experimentally. You should have some a priori reason for trying a method. So, for example, the previous ones, they were using language-based models, but there is some rhyme to it in the sense that those language-based model try to capture. So they try to capture sort of correlations across several length scales, which is true, which happens to give language its complexity, but it's reasonable to suggest that that also gives these big molecules complexity. So as I said, I'll talk about coarse graining here. Again, coarse graining is something that was mentioned before, but what is it useful for in this context? So this is again the structure I showed you earlier. So this is the backbone. But there's so many of these other atoms hanging around. So if you wanted to do this other problem of trying to see how you get to the structure, and you want to and you started doing an all atom simulation on this, that would be too slow. So what is often done is that you focus on a few atoms, and these might not even be real atoms. These might just be some center of mass type objects, and you only take those and you try to simulate just with those and with some potential functions that are geared to just see how these virtual atoms evolve. And then that it loses information, but it can be faster. And so one thing we would want to do is to use the structure prediction. So this is what we want to do. So we have not done anything so far. So that's another thing I should have said. So it's more about what we want to do now and what we've been exploring. So there is structure prediction, there's better coarse grain simulation, but they're related problems. In the sense that the predictive structure, these methods for predicting structures can help you whittle down what are the correct coarse grainings in some ways, and they can also let you figure out what should be the starting configuration in some sense, where you should start. I mean, so these structure predictions are approximate, so you could try to even make them better by running a simulation from them. So that was all I was going to say about here. And are there any questions? If there are no questions, I'll ask a very embarrassing question myself. So, so that's okay. Yeah.
Are you trying to figure out the representation, like what should be coarse grained based on some machine learning, or are you just using the structure to get?
So right now, so the one thing we would want to just do is to go between coarse and fine grained using learning techniques. So the learning technique does not come into figuring out what the coarse graining is. I mean, it helps it in the sense that if you know the structure predictor, it tells you what the angle should be and so on. But after that, the main problem would be to go between coarse and fine grained structures.
And that's currently not a learning object.
So yeah, so people have done things which will look like learning, like clustering and so on, but not with networks.
So RNA structure is dependent on basically size of the RNA as well. So are you trying to look at diverse sizes?
Yes. So as I said, like there are only about 6,000 sequences.
Correct, yeah.
And so for example, in this one, the result that I mentioned here, yeah, this one. So I said that their median length is about 80, and their highest length sequence was about 400 something. So that. And the reason I think, if I recall correctly, the reason the larger ones were discarded is because parts of them were very similar to the things they already had. So it would just bias the learning. So these kind of problems are also there about because the data is so less that you cannot do many other things.
Well, I'm going to show you in the smaller data set later.
Sorry?
Smaller data set than yours. I'm going to show you in smaller data.
Oh, I see. Okay. So maybe I should ask. Sorry. Yeah. Okay.